We're just gonna wait a couple more minutes to allow people to join. We'll begin in a minute. There's still a lot of people joining. All right, everyone. I guess we shall begin. I don't want to hold it that one up. We have a very tight schedule today. So hello, everyone. Depending on where you are, good morning, good afternoon, and good evening. My name is Emmanuel Ho, and I'm an associate professor at the University of Waterloo. I am also a member of the Waterloo Institute for Nanotechnology and one of the co-chairs for today's event. So before we begin, I would like to state that the University of Waterloo acknowledges that much of our work takes place on the traditional territory of the Neutral and the Shinabeg and Haudenosaunee peoples. Our main campus is situated on the Haldimand Tract, the land granted to the 6 nations that includes 6 miles on each side of the Grand River. Our active work towards reconciliation takes place across our campuses through research, learning, teaching, and community building, and is centralized within the Office of Indigenous Relations. I would like to welcome all of you to the Network for Sustainable Nanotechnology Global Summit virtual launch event. The theme for this event is Nanotechnology for a Healthier and Sustainable Future, which is in support of the United Nations Sustainable Development Goal number 3, that's focused on good health and well-being for all. The event is divided into 2 days. On day 1, which is today's session, we have 2 amazing keynote talks along with a panel discussion focused on digital health and artificial intelligence diagnostics. Tomorrow, day 2 of our event, the session will be focused on nanomedicine. Before we begin, I would like to simply state some simple housekeeping rules. Please kindly keep all of your microphones muted and your cameras off at all times. If you have any questions, feel free to ask them in the chat box. In addition, you may also use the raise hand function in Zoom to allow the moderator be aware that you have a question that you would like to ask. Please do keep in mind, though, due to the interest of time, we may not be able to address all of your questions. So feel free to email the speakers and the panel members at the end of the event, and I'm sure they'll be happy to respond to any inquiries that you may have. So to begin the session, I would like to introduce Dr. Sushanta Mitra, Executive Director of the Waterloo Institute for Nanotechnology, to say a few words. Sushanta.
Thank you, Emmanuel. Thank you, everyone, for joining in. It's such a pleasure to be part of this exciting journey. When we started the Network for Sustainable Nanotechnology, it was quite a nice way to bring together global leaders across different parts of the world with a common focus to see how we are able to create a better future in terms using nanotechnology for sustainable society. And really, we are fortunate to have our members at the Waterloo Institute for Nanotechnology at the University of Waterloo, and similarly Sydney Nano at the University of Sydney, to work together to create this exciting program. This is a launch event, as Emmanuel mentioned, and the actual summit will also take place in the summer 2022, hopefully in person. Let's keep our fingers crossed, and thanks everyone for joining in today. Thank you.
Thank you so much, Sushanta. Next, I would like to introduce Dr. Charmaine Dean, Vice President, Research and International at the University of Waterloo, to say a few words.
Welcome, everyone. So great to have everyone here attending the Global Summit launch event. And as Sushanta says, it's over the next 2 days, but it's really a teaser for the Global Summit in June. I'd like to really thank Sushanta Mitra and his team at the Waterloo Institute for Nanotechnology, and of course our partner at Sydney Nano at the University of Sydney for organizing the event. As Emmanuel says, a really interesting focus given the current state of the world, nanotechnology for a healthier, sustainable future. We all know about the Sustainable Development Goals created in 2015 to address some urgent issues, the environmental, political, economic challenges that are impacting the world today. And it's really very, very helpful to see the Waterloo Institute for Nanotechnology focusing their work and linking them by mapping the thematic areas to the Sustainable Development Goals. I don't know that we have many organizations at the institution that actually does this mapping and really focuses on the SDGs, and having the researchers in the institute also engaging in sustainable projects that benefit our Indigenous communities. That's really, really very welcome. We also, of course, value global partnerships, and I'm also the Vice President International, so I'm really thrilled doubly that we have innovations in research linking to the International Network for Sustainable Nanotechnology. So Australia, the Netherlands, the United States, Japan, all promoting nanotechnology advancement and application. I'll also note that the pandemic years have been very difficult and challenging, and they've also shown us that we live in an interconnected world, a world that's really important for us right now to build upon navigating challenges and succeeding despite adversities. And I'll also note that the pandemic has also shown us all the inequities that are really at play in the world. And inequities related to health are substantial. So another reason why we should be focusing and driving the importance of Sustainable Development Goals. And another reason why we need to collaborate to solve problems and build innovation that will lead us to navigate large-scale international crises as we've just faced. I encourage you all to collaborate over the next few days. I hope that it's so unfortunate that all our conferences have to be held virtually. I hope that the platform provides you the opportunity to still collaborate in interesting ways. And I noted that Sushanta was very optimistic that we will be able to meet in person in June. Well, that would be wonderful. And we all look forward to seeing that happen. Thanks, Emmanuel, and thanks to the team organizing this event.
Thank you very much, Charmaine. Next, I would like to take this opportunity to introduce the moderator for today's session, Dr. Rohinton Medara, President for the Center for International Governance Innovation.
Emmanuel, thank you. My job is a simple one. It's a privilege to be with you at your launch event for an exciting network topic. As the program mentions, we have two keynote speakers who will speak for about 15 minutes. We then have given each one 5 minutes for a Q&A. So if you have questions, by all means, send them in. And in the interest of time, I might sometimes combine them. We'll then take a break and then come back with a third panelist and have a moderated session to round off the morning or evening, as the case might be. So thank you again all, welcome. And I'd like to begin by asking Professor Dean Ho from the National University of Singapore, Department of Biomedical Engineering, to speak with us for about 15 minutes. I'm not going to go into great detail on bios because you have them in your program and in the interests of time, but Dean, welcome.
Thank you so much for having me. Hopefully everybody can see my slides okay and hear me okay. Great. Thanks to the organizers for the kind invitation. It's great to see so many friends in the audience today. Hopefully we'll be able to get together soon. Today I'm going to talk a little bit about this nexus of nanomedicine and digital medicine and how lessons learned from our work in nano has eventually helped to guide how we are now optimizing clinical treatment for things like combination therapy, dosing, N-of-1 medicine, and beyond. And so I'm just going to give a brief introduction to the institute that I direct, called the Institute for Digital Medicine, because I think this helps to define how we bridge ideation with deployment of emerging technology at an interventional scale. So about 99% of what we do at the Institute for Digital Medicine, as well as the second institute that I direct, known as N1, is interventional, is treatment related. And it takes a team to do this with an ethos that technology alone cannot transform healthcare. And the expertise represented in this institute spans nano, digital medicine and AI, behavioral sciences, implementation, reimbursement, and beyond. And so we have faculty members from engineering, from the clinic. My deputy director is a surgeon, as well as an AI expert, wonderful nanoscientists like Dr. Min Lei, and as well as people who work in the business school, who specialize in operations, analytics, incentives development for adherence for our patients, as well as behavioral scientists to help us gauge what the barriers are to true adoption of new treatment-related technologies in the clinic. In fact, the first hire we made out of Wisdom is not an academic. He actually hails from the payer side. He's from insurance with expertise in medtech. So it really takes a team with the mindset that it takes a lot more than technology to truly transform healthcare that allows us to do what we do. And that has resulted in a rapid upstart of a number of prospective interventional trials for everything from liver transplant to digital therapeutics, to nanomedicine, all the way to using video gaming to address brain cancer patients. And so this is just a snapshot of some of our trials underway. And again, I think that that's owing to a lot of the broad expertise that we have in our team. And so I want to talk now, starting with nanomedicine, and how we took lessons learned from nano into optimizing patient care. And when we think about the key objectives of nanomedicine, we think about often three key areas, right? How do we deliver the right drug, the right dose at the right time? But then I think what we've learned has since allowed us to ask deeper questions on how to be more interventional, to define what happens at the cellular, tissue, and ultimately patient level. So if we want to deliver the right drugs, we have to ask ourselves the question, how do we pick the right drugs? Do we use mechanism of action, which is how drug discovery and development is largely done now? Is our goal to achieve synergy? Because as we'll learn, using different approaches outside of mechanism of action to leverage unforeseen interactions, unforeseen off-target effect to optimize treatment, we may need to look beyond synergy as well. And I'll talk about it. Right dose. So if we want to give the right dose, what is the right dose? Is the goal to achieve maximum tolerated dose or to achieve threshold levels of drug at a disease site? Because if I were to then say, for some patients, the more drug you give, the worse off they are. And the less drug you give, the better off they are in terms of efficacy, not just toxicity. It allows us to rethink this statement. And the right time. What is the right time? Do we have to rethink scheduling? And do we have to rethink targeting? So lessons learned has allowed us to ask questions to think or to reimagine how we address these key challenges. So I'm going to quickly go through some of the nano that we've done and then go to lessons learned. So many years ago, we started working on nanodiamonds for everything from MRI contrast agents with Tom Mead at Northwestern, to drug delivery with Ed Chow, who is at UCSF, now at NUS, and many other collaborators, all the way to making contact lenses laced with nanodiamond to do triggered drug release for glaucoma. But then eventually all these lessons learned led us to how to optimize combination therapy with nano. And so again, the talk is how do we take lessons learned and then go towards bridging the nano lessons learned with clinical intervention. So with nanomedicine, our teams around the world with nanodiamond, other nanocarbons, gold nanoparticles, polymer nanoparticles, and of course, with lipid nanoparticles, have looked at improving efficacy and safety with nanoparticles. And you can see here with free drug and nano drug, we can do better. With targeted nanoparticles and nanodiamonds, we can cause regression when we target with nanodiamond. Moving on, we look at things like safety and toxicity. So we conducted non-human primate studies of nanodiamond safety, and no worries, I'm not going to go over all this information, but suffice it to say the nanodiamonds are pretty well tolerated. We recently completed a canine PK ADME serum urine analysis study on one of our nanodiamond drug particles, and you can see that free drug is not so great, and when you very simply combine the drugs with nanodiamonds, things get a lot better. And this was dual gender, larger number of animals, fairly comprehensive PK ADME, and we can see that nanodiamond's pretty well tolerated. A few years ago, we advanced this all the way to the clinic, where we developed a nanodiamond composite for root canals, both both drug containing and not drug containing. We have not yet initiated the drug containing study, though it was cleared for start. We're going to actually try to scale that trial up and run it here at NUS, and work with UCLA as a multi-site. But at the same time, we were able to conduct a nanodiamond only study as a first in human to look at things like, could we improve the clinician's ability to address the root and fill the root? As this study went on, we found that the diamonds were well tolerated, the root canals performed well. And so this was a nice first step towards bridging cell to tissue to animal, large animal, all the way to clinical studies. But eventually, we started thinking about how to, like many teams around the world, can we deliver more with nanoparticles? Can we deliver multiple drugs? Can we target better? And so we were confronted with a challenge that we wanted to work towards, which is how do we know which drugs to combine? So this work was done by some of our colleagues in Europe, where they looked at delivering monotherapies in animal for a solid cancer model. And you can see here, the large number here is the drug number, and then the little sub number here is a dilution factor. So it's like a dose. And you can see here, drugs 4, 8, and 9 at whatever doses didn't do anything for this tumor. All right. So if you then start thinking about combining them, so if you look at the control saline, tumor gets worse. But if you look at combo 4 sub 1, 8 sub 2, 4 sub 1, 8 sub 2, drugs alone don't do anything. Drugs together don't do anything. All right. But then if you look at combo 6, which is drugs 4 sub 2, 8 sub 2, 9 sub 4, 4 sub 2, 8 sub 2, 9 sub 4 don't do anything alone. But when you combine them, they actually do something, which then led us to wonder, hey, are we missing efficacy with drugs? Because if you purely look at single drug screening, nothing happens. But if you combine them properly at the right respective dose, things can happen. Right. So let's try to tackle that first problem of delivering the right drugs. But that's a challenge because we know that when developing drugs, especially under pandemic conditions, there's a lot of issues. Take too long to develop, low success rates. And then the big challenge is what drugs do you combine at which respective doses? All right. We often think about, look, let's attack multiple mechanisms and then just dose escalate. Right. But the issue here is you actually have to simultaneously find the right drugs and the right doses, because that's what leads to this top-down approach to come up with truly optimized combinations, right? And there's another problem. Even if you take good drugs, but you give them at the wrong dose, you may get no efficacy at all. And what happens there is it creates a misperception that these drugs cannot work at all for patients. And I'll show you an example later of a patient where we did not change the drugs. There appeared to be minimal to no efficacy at clinical dose for these two drugs. But without changing the drugs and just by changing the dose with a small amount of data for this patient, we actually acquired efficacy. Don't change the drugs, just reduce the dose and they go from not responding to responding. So all of these issues create this massive parameter space that we have to resolve, and areas like digital and AI can help. Right? So I published this paper about 2 years ago looking at AI in cancer therapy. And when we think about AI, when we think about cancer therapy, when we think about nano, we think about, hey, can we discover better drugs? Can we better design materials at the nanoscale? And of course, the answer is yes. A lot of exciting work. But after the candidates are discovered, right? Pharma right now is very discovery driven, meaning if you come up with a great compound, you should be able to clear trials and improve outcomes. But as we know, many compounds fail. And so we have to think beyond sensitivity. We have to think beyond just finding high doses, and we have to better design combos and then better dose those combos. Right, so I'm going to give a quick cartoon here to illustrate what I'm talking about, right? So oftentimes we get approached by teams to say, hey, look, if I give you data on patients for a 2-drug, a 3-drug, or a 4-drug combo, right? Can you use AI to predict for me what the best combo is or what I should give the next person that walks in the room? And what they'll often say is, I've got millions of data points at fixed dose. Can you predict? And we can say, well, even without using AI, we can probably try to find out what combos performed better. But let's make things more interesting. Let's say we got a much larger pool of drugs and someone comes to us and says, hey, look, can you find for me what the best possible combo is on a liver cancer model with 12 possible drugs? But the problem here is if we know that dose matters too, it's not just about picking the drugs and then looking for the dose. We actually have to resolve both, because if you know how to optimize dose, the drugs that comprise the combo you think you're going to need may be an unforeseen combo. Let's make it more interesting. Let's say drug number 5 doesn't even work on its own. We tend to kick it out, but let's leave it in, right? But again, that creates huge parameter space. So how do we solve this? So instead of taking huge amounts of data, we actually prospectively acquire less data, but at specific permutations to cover that whole parameter space. And from this prospectively acquired small data, we can then truly find what the best possible combos are, all the way down to the worst possible combos. It's not enough to run these small experiments. We run this quadratic optimization process, which I'll get into a little bit, which means we can reduce the number of experiments we need, but we need specific experiments. And then after this AI discovered relationship, we can quickly find what the best possible combos are. And we ultimately applied that to nanomedicine. Published this paper about 6 or 7 years ago, where with nano diamond modified drugs, unmodified drugs combined, we were able to find which drugs, nano and non-nano, at which respective doses resulted in the optimal outcomes of efficacy and safety. And I will add that this is applicable at both in vitro, in vivo, and clinical stages. So the first use case I'm going to give is a COVID-19 use case, which is a study we initiated in 2020 to find the best possible combos by crowdsourcing the live virus. Both wild type, beta, delta. We're actually starting the Omicron study right now. And from this large pool of drugs with feedback from clinicians from around the world, we were able to rapidly pinpoint unexpected drug combos, some of which have been cleared for trial start. Right. And again, our optimization work is not merely a single combo study. It's multiple combinations that are mapped out from this prospectively driven study. So doctors can come to us and say, hey, I need your best 2-drug combo. I need your best 3-drug combo. What's your best 2-drug combo without, Paxlovid? Or what's your best 5-drug combo without? These are things that we can explore using this approach. All right. With the minute or two that I have left, I'm going to quickly go through a couple of other examples where we optimize the dose. We ran this study with UCLA where we were optimizing transplant patient doses. And you can see this is a single patient over 4 weeks. and their drug interactions and how dynamic this is. And these are between patients, how different they are from each other when their drugs interact at a specific point in time. But I will say we discharged our optimized dosing patients about a month earlier compared to standard of care. Not long after that, we were approached by a prostate cancer patient who was responding to care at 2 drugs, but it was highly toxic. It was considering dropping the trial. But with a small amount of data from just this patient, we don't collect large amounts of data, train algorithms to then treat the next patient. We use only a patient's own data to manage only their own care. We found that by some modulated dosing and reducing the dose of a single drug by 50%, they could actually increase their efficacy. Imagine reducing the dose to increase efficacy. We further translated that into thinking about for another set of patients. This was a gastric cancer patient who was not responding at all to any treatment, right? So they were thinking this was end of care, end of life treatment. And by actually slowly ramping up dosing to calibrate their response, we actually found efficacy for this patient at low dose. The final patient I'll mention is another GI cancer patient who is not responding to care, which is standard. It's always encountered in oncology, where you just move on to another regimen after, right? If a person doesn't respond, you move on until you run out of options. But because we had prospectively calibrated this patient with variable dosing and corresponding efficacy with serum markers, we were able to see that if they had dropped the dose at the right time for this patient, you could actually switch this patient from becoming a non-responder to a responder. And that's exactly what happened, right? Imagine going from not responding to responding by dropping the dose, right? Better quality of life, suddenly responding to care. We've since started correlating our dose reductions and our reduction in readmissions due to toxicity with savings. And for some of these patients, we were saving them thousands of dollars a month by using the AI to guide their treatment. Last slide. What's next? We've got a lot of trials underway. We have more trials underway with both optimized combinations as well as optimized dosing. We've made a lot of our information available online for clinicians to determine which drug combos may be actionable and not actionable for things like COVID. With the help of many friends and colleagues in the field, including some in the audience today, we put out a comic book on AI for infectious diseases, which is available online as well for free download, as well as the libraries in Singapore. And some of the work that I do with WHO and a number of philanthropic organizations to continue our work in COVID, we're very grateful for. So again, I wanted to acknowledge everybody who's helped drive this work and the funding agencies that support this work. Thank you for having me today, to the organizers, and happy to take some questions.
Great, Dean, thank you. That's a great way to kick off our discussion, and you've given new meaning to the phrase less is more. And it's something we might come back to in the panel as well. We have about 5 minutes if someone wants to raise their hand or send in a question. And while people do that, maybe let me just ask you, Dean, you're located, it might seem like a naive question, but since we're talking about data and AI, you're located in a small country. How do you do your clinical trials, and what kinds of conventions do you have to follow for cross-country data?
That's a great question. So clinical trial innovation is actually one of the arms of Wisdom. And we have a team of clinical trial designers that help us do what we do. Because our trials are N of 1 trial. So N1, the other institute I direct, is for that specific reason. We're purely clinical, and we design trials for every patient that we enroll. But these trials are N of 1 trials, but multi-patient, which means while we individualize their dosing using only their data, the reason we do that is so that we can, because the model doesn't change, right? We don't take 100 patients, 1,000 patients worth of data, train an algorithm, make a prediction on dosing off that algorithm, and then treat the next person that walks in the room, right? We don't do that because certainly with this data, it's, there's issues of data bias, of data completeness, et cetera. We prospectively calibrate each patient. So what that means is a patient comes in, we have a cleared study where we give them a depending on what the clinician prefers, high dose, middle dose, low dose, if it's more aggressive, low dose, middle dose, high dose, low, high, medium, as long as we give them a range of variable dosing, all within safe limits, and then measure their corresponding efficacy at each of those doses, we're able to plot out this digital avatar, if you will, for each patient, defined by their unique constants. If you then take each patient's constants, and then look at it from a population or a multi-patient viewpoint, you actually get better insight into how to dose populations as well, right? So in Singapore, there may be less patients, but the info we get from each patient is a lot more. It's also longitudinal, which can then be used to expand into the region. And we also have run trials before and with other institutions in other parts of the world as well. We've remotely run trials. And so in terms of cross-border data usage and guidance, that's the thing. We actually prospectively acquire the data. So we don't really collect tons of data from across the border, except to do some analysis as a POC for the clinical teams before we initiate prospective trials. And those, of course, are approved by an efficient but rigorous clearance process.
Okay, we have time, I think, for one more question, and then we'll go to Kishor Wasan.
Oh, thanks, Dean. I want to ask you, with drug repurposing, there's a lot of conversations at the moment with drug repurposing. There's a lot of work in this space. Can you comment whether the approach which you have could actually help with drug repurposing?
It's a fantastic question. Yes, and I'll give one example. So when we think about repurposing, we think about, can we take a drug developed for something else and apply it to something else? Can it be used for cancer? Can it be used for COVID? And the way we do repurposing, especially with AI right now, is centered around modeling targets, right? Like, hey, here's a pathogen that's just emerged. Can we model the target and see what else can intervene? For cancer, can a diabetes drug block this target to help with cancer? I think we have to think very differently. Repurposing and optimized repurposing are two totally different things. Repurposing is very much like traditional drug discovery, just without the new molecule being built. And when we do that, it seems very clear that if you block one target, if you block two targets, efficacy should be better. And that may very well be the case, but we're not asking that question. We are asking, from a bigger pool of drugs, how do we find the best possible set of drugs to combine? And that will often go beyond pure mechanism of action targeting. We're leveraging off target, we're leveraging unforeseen drug interactions. And then we get the question, well, how dependable is your disease model? Is it purely in vitro? Is it ex vivo? Is it preclinical? But we come back with the same question, regardless of what model you use, and we've used them all, right? You want to come in with an optimization perspective, not merely a bottom up where you say, look, let's combine these two drugs, because synergy is not enough, okay? And by the way, synergy is dynamic, okay? It's not fixed, right? And so the final thing I'll say is, we get patients, right? We have Ed Chow on our team who's leading a trial. We get patients who come in who have failed 6 lines of therapy. They're going straight to hospice care within a few weeks. We get a biopsy, and a biopsy, you don't have a lot of sample. Okay, so in traditional repurposing, you do a couple of single drug assays, maybe some known combos with the small amount of tissue you have. And then you say, look, out of these 10 experiments, this drug is the best. It's a drug sensitivity test. You give the patient the drug, it probably won't work out very well, okay? We do something different. We take the sample, dissociate, and as I mentioned, we don't need a lot of experiments. So instead of just drug sensitivity, we run prospectively these different permutations of drugs picked with clinicians. And instead of testing 10 different combos out or 10 different options, our one experiment driven by real data and then optimized gives us combos ranked from number 1 all the way down to 10,000. The clinicians can look and say, oh, you know what? Let's not just take combo number 1. Let's first look and see all the combos they failed, see how they rank to build confidence in this experiment. And they're like, yeah, they actually rank pretty low. We don't like combo number 1 because it's 6 drugs, okay? But we like combo number 3 because it's 2 drugs. They're repurposed, they're safe, we're willing to give these drugs. And patients who are days away, weeks away from hospice care have achieved complete remission, right, from doing this. It's not just the remission, it's high positive predictivity, high negative predictivity as well. We can also tell clinicians which combos to not give. And we do that for COVID, we do that for oncology, and this is prospective here, right? And so it's not just about traditional repurposing, it's optimized repurposing. It's a very different strategy. Thanks very much.
Great. Look, we'll come back to some of these points, but for now, Dean, let's move to Dr. Kishor Wasan, who is with Skymount Medical and the University of British Columbia. Same thing, Kishor, about 15 minutes and then 5 minutes for questions.
Okay. Can you guys see my slides okay? I'm having all right, let me try again. Oh boy. All right, let's try again and you tell me if you can see it.
Yes, but not the full screen yet.
I'm waiting to do that. Okay.
Yeah, that's fine.
Yep, good.
All right. Well, thank you very much, everyone, for having me. And again, I want to thank the organizing committee as well as particularly Dr. Manuel Ho for inviting me. And it's a pleasure and honor. I don't know many of the people on here, though I did notice that a number of you actually joined my LinkedIn page today. So thank you very much for doing that. My name is Kishor Wasan. I'm a distinguished university scholar, professor, and co-director of the Neglected Global Diseases at UBC. But today I'm going to wear a different hat. I work as the chief medical and scientific officer at a small medical research group out of Calgary, though we have sites in Louisiana State University, as well as folks in California and in Washington, D.C. And what we're going to talk about today in a short period of time is how our use of artificial intelligence and machine learning in discovery and development of novel therapeutics for emerging infectious diseases. But I must say that building on my esteemed colleagues' previous talk, we too have ventured into cancer chemotherapy. We're looking at Alzheimer's and so on. But we're going to use the example of COVID-19 because, you know, it's been 2 years, ladies and gentlemen. And, you know, what has happened is everyone was doing research in another area. And then when COVID hit, everyone pivoted their research to try to address the issues with COVID-19. So I'm going to use COVID-19 as an example of the power of our algorithm and AI, how it's got us to the stage where we're actually doing human studies in California and we're embarking on studies overseas as well. You can't do anything without collaborators and so on. And at the very end, I'll talk about all the list of our partners that we have. But on the far left of the screen there, Dr. Supratik Mudakudapadiye is the brilliant genius behind our algorithm called Deep Drug. Chris Galliano is been outstanding in his work from the military perspective and brings a military background and naval warfare systems background, which is very important and gives a unique thing. And then there's good old me. So Skymount Medical, what happened is that they worked with Louisiana State University and with their Deep Drug renowned AI platform. And this platform is to advance drug discovery and development in a very short period of time. And Deep Drug has actually gotten some really cool accolades in that it was a semi-finalist for the IBM Watson Artificial X Prize a couple of years ago, and was really the only medical AI that made it this far. And it's because of some of the unique things that Deep Drug has put together. And so, of course, as the previous speaker has mentioned, we're all really after the same thing. Are there ways that we could use artificial intelligence to shorten the times of R&D cycles, to improve successes, make better decision making, and ultimately get us to a place where we have a cost-effective therapy in a much faster period of time? And can AI be an incredible tool to do that? Now, I've got to tell you, I'm a clinical pharmacologist and pharmaceutical scientist for over 35 years, and and I've done it old school. I had a big old lab at UBC and did it the old school way. And I have to tell you, if this algorithm was around 10 or 15 years ago, I would have been a long time out of business. And so it's really exciting to see this kind of algorithm applied to help us at the end of the day get greater patient therapy. So how does deep drug inaction work? And again, as I said, I'm going to use the COVID-19 since it's timely and that's what everyone is talking about as an example of how we apply deep drug to get combinations. And by the way, someone asked about repurposing. We have not only new chemical entities, but we also have repurposed drugs that we're actually in the clinic right now because of deep drug. And I do agree. I mean, I'm a pharmacokineticist and drug therapy guy myself, and I agree with my colleague who just spoke about the optimization of dosing and so on and so forth. Could give you a whole huge talk on that, but because of the length of time, we won't do that. So how does deep drug work? So what deep drug does is it identifies the human proteins that the SARS-CoV-2 virus uses for entry, replication, transmission, and fusion into a cell. And then what it does is the AI maps known antiviral peptides to these different cell mechanisms by analyzing protein to protein interactions and ranks its effectiveness. And then what it does is it recognizes similarities between these antiviral peptides and existing drugs or a whole pool of drugs that are in databases. This is the systems that are used, that it's really a multi-complex system that is used to be able to decipher that. We've just published a paper in Drug Discovery Today at the end of last year that goes not only into these systems more in more thorough ways, but also compares to the other AI systems that we see as competitors and how ours differentiate it. The one thing I don't mention here that's not in this slide is we've also brought in pharmacokinetic, pharmaceutical science, and pharmacology principles now into our deep drug algorithm. So not only drug-drug interactions, but ADME principles, so absorption, distribution, metabolism, and excretion of drugs, drug toxicity, drug formulation, as well as physical chemical properties of drug candidates, using, for instance, the Lipinski rule of 5. And we've integrated all of those aspects into our analysis to be able to now have predictive drug candidates that we can spit out that we can then do for further development. So the other way that this all works, I'm just going to flip through this part because, is what happens is that we identify effective building drugs in known drug targets, and we develop chemically sound derivatives for building blocks and filter out toxicities as well as specificity. In this case, it could be bacterial facility. The thing that you need to keep in mind here is the fact that not only can we work with existing drugs and existing candidates, but we can build new drugs and build new chemical entities. But for expediency purposes, I think for COVID, as someone asked earlier, we went to a repurposing strategy with drugs simply because we were trying to find something to help patients now. Here's some of the recent publications that Deep Drug has had, and you can click on it, and I'm sure these slides are available. And the Drug Discovery Today paper was just published at the end of last year. So what have we done? We've taken over 1.5 million drugs. By the way, we can do trillions of combinations and different combinations and so on, just like our predecessor colleague had talked about. And what we did is we did antiviral computations of these 1.5 million drugs and came up with lead combinations. Then what we did is we really set up a classic drug discovery and development program at that point. So we set up cell culture. We looked at lung cells. We looked at kidney cells infected with SARS-CoV-2. And of course, we used the Washington strain and the Canadian strain. And just like my colleague had mentioned, we are actually currently doing studies right now with Omicron, actually both variants of Omicron as well as Delta, to see if our drug combinations and our monotherapies hold up in cell culture like these. And then what we did is we took a few leads. And in our case, we were really trying to get something quickly into patients and going after a very targeted situation. So we have an in vivo mouse study, which is the transgenic mouse model for the ACE2 protein. We infected them with a severe infection of COVID-19. And in fact, remdesivir only worked about 20% in these animals, and the majority of animals die within the first 5 or 6 days. And then we looked at the results from that. And then based on all of those, we decided to go into human studies. And for us, our target human population is a little bit different than the Pfizer compound or the Merck compound. And of course, it's complementary to vaccines. We were looking at very, very early stage patients that have mild to moderate COVID that we could give right away these drug therapies to try to decrease the severity and length of the disease and to prevent what we consider long hauling syndrome. So this is very early stage, very early on, and it's kind of a niche in between the standard of care that's happening for patients for COVID right now. We've got wonderful vaccines, we've got some interesting oral therapeutics for more later stage, but what's happening for earlier stage? There really are no oral medications out there for that. And we were able to come up with a couple of combinations as well as a couple of monotherapies that were very safe, that were very effective. And it's very interesting what was mentioned previously. We actually did some dosing as well and actually found that actually lower doses of the two gave us a better effect. So we didn't have the issue of toxicity as well. And it's just a summary of the work that we just discussed here. And some really exciting news, actually, we just announced today. We actually have a human pilot study going on at UC Riverside in California, recruiting and enrolling patients as we speak. But today, we just formally announced that we are going to be doing a clinical trial, a large clinical trial in the UK. The MHRA in the UK, which is the FDA equivalent, the United States of the MHRA in the UK, has given us formal approval to move forward on a clinical trial. We are also looking to do a clinical study in Ukraine, and we're actually waiting for Ministry of Health approval there. But as everyone is probably knowing, Ukraine is heavily in the news today, and we're hoping that we're going to be able to do our clinical study there, and hopefully geopolitical situation doesn't get us into trouble there. So the next slides, I want to show you very interesting some data. Data is really important. So we've now generated 4 patents within a year. What I show you on the y-axis here is both in kidney cells and in renal and in lung cells, the infection of COV followed by treatment for 48 hours. And you can see that these compounds were very, very potent in a short period of time. The lead compounds and combinations that we brought forward from the AI that we did in cell culture, we had greater than about 75 to 80% showing actual effect at non-toxic levels. So that was really exciting because what that was saying is that our AI was predicting 7 to 8 out of 10 drugs and combinations that there might be something there. This is actually further work of one of our combinations called SM19. On the y-axis, I'm showing, and this is, by the way, unpublished data. I'm showing you fresh, brand new data. On the y-axis, I'm showing you the percentage of inhibition of the SARS-CoV-2, and in the lighter text is the inhibition of cell viability measured by MTT. And on the y-axis is the dosing, and you can see that we're able to inhibit the replication of the virus at non-toxic doses. And this is our monotherapy that was done in lung kidney cells, in lung carcinoma kidney cells. Again, on the y-axis, we're showing the inhibition of the SARS-CoV-2, and then in the red is the inhibition of cell viability or toxicity. And again, at non-toxic doses, we were able to show a significant reduction in inhibition. And that led to, this is also some additional work as well on live virus and TCID50 of another compound, as well as inhibition of SARS-CoV-2. But this led to our animal studies, and I haven't been able to show all the treatment groups here, but we did use remdesivir as a positive control, and we had a number of other combinations and monotherapies as well. But what we're showing in a very, very severe infection of COVID, not the target we would actually do in patients, that SM4 and SM19 almost had 40% reduction in the viral load in less than 5 days. And in fact, 70% of the animals in these groups actually had greater than 90% reduction in the viral load. These animals survived, and in fact, their body weights, which kind of went down, actually got back, were going back up again. In the untreated control, all these animals died within that first 5 days. And so it showed that our combinations and our monotherapy were very effective in animals. We also did renal function, liver function, biochemical analysis, and found that these were at non-toxic doses and non-toxic combinations. And that led us now toward the clinical studies that I've talked about today. But we've also ventured into nutraceuticals. There's been a lot of work, as you know, with vitamin D, with zinc, and some of these other nutraceuticals. So we also had predicted a number of nutraceuticals that would be maybe potentially effective against COVID. And we show 2 of them here. But we also know that these nutraceuticals are very effective in immune supplementation, in cardiovascular health, in antioxidant, as an antioxidant, anti-inflammatory. And one of the side effects of COVID-19 is vitamin and mineral imbalance, secondary to the COVID infection. And we had lots of people coming into pharmacies and wanting multivitamins, going off the shelf like crazy, looking for that immune support and cardiovascular support, and that rebalance of vitamins and minerals. And so from that, we developed, using our deep drug outputs, a novel nutraceutical that we call Inhibitol. And so Inhibitol is now in the United States. Is marketed as not as a COVID treatment, because you have to do proper clinical studies and proper studies so it would be a pharmaceutical, but we are marketing it as a nutraceutical with immune support, cardiovascular support, and antioxidant. And we see this as a complement that we can give to all patients. It doesn't have to necessarily be COVID patients, but patients that are looking for that vitamin mineral imbalance that is secondary to a lot of emerging infectious diseases. So the take-home message here is this, is that in a very short period of time, we brought together a number of people together from different branches, from different aspects of industry, academia, CROs, government, and so on, and really put together the SkyMount medical team. Really to not only to deal with COVID-19, we were all off doing other things and other research, but also to take advantage of this great deep drug AI technology and apply it not only for SARS-CoV-2, but into the future for other diseases and for other treatments as well. And this is just a decision tree that we use at SkyMount to determine how we came to the leads that we have today. And like I said, it's a village. It takes a number of different groups and organizations to work together, and funding from these groups to help us. LSU is the hub where deep drug was discovered, and it's one. UBC, where I'm at, we're working with our Neglected Global Diseases Initiative with the folks at SkyMount. Vito Intervac in Saskatoon, which is related to the University of Saskatchewan, is a world leader on vaccines, and in fact has several vaccines in clinical trials for COVID and for many other diseases. We're doing clinical studies at UC Riverside. We're working with a outstanding formulation group out of Vancouver, led by Dr. Marcel Bally. And in fact, Dr. Peter Cullis, who's at UBC, was the gentleman that discovered the lipid nanoparticle that has been used in the Pfizer vaccine. We are actually working with Peter and Marcel and some of the work that we're doing with the AI. SafeBridge is an outstanding organization that does clinical pharmacology and toxicology and so on and so forth. So with that, thank you very much for your time and your attention, and sorry that I had to talk so fast. Thank you.
No worries, and we do have more time after the break as well, Kishor, to come back to some of the points we made, and really good luck with your Ukraine study at this point.
Yeah. We've invested 6 months in Ukraine, and 2 of our senior leadership just got out of the country literally a few weeks ago. And in fact, we're talking to Ukraine daily. And one of our people we're working with actually had to move his family from Kiev to Lviv, the western city in Ukraine. So it is a very scary time for individuals.
we hope we don't see you on CNN is all I can say. Look, we still, if anyone has a burning question, raise your hand now for Kishor. And if not, we're going to go into a 10-minute break and come back with our third panelist and have a discussion that's going to sort of cross over into Dean and Kishor's very many good points. So does anyone have a pressing question? Lots of applause for you, Kishor. So let's leave it at that for now. Emmanuel, do you want to say anything, or should we just go into our 10-minute break?
Let's go into our 10-minute break, and then, I'm looking forward to the panel discussion.
Okay, good. Okay, so if we're all back, I'd like to get going with the second half of our program today. And for this bit, Dean Ho and Kishor Wasan will stay with us, and we're going to be joined by Professor Mohammed Kohandel from the Department of Applied Mathematics at the University of Waterloo. Is Mohammed with us? Do we know? Oh, there you are. Hi there. Good. And I believe if you have questions, by all means, put them in the chat. Emmanuel and I are following the chat, and then we can sort of have a group discussion based on the things you would like to hear from our panelists about. I'd like to begin by asking each of you something that someone raised previously in the chat, and I think it applies to them all, which is, it's a sort of narrower version of the question that both of you talked about, which is AI kind of adds octane to the speed and the thoroughness with which nanotechnology can be applied. On the other hand, you seem to be using relatively modest data sets, while the principles of machine learning and AI seem to require lots and lots and lots of data. And I'm wondering if you could talk a bit about the quality and quantity of data you use and how it alters your work. And maybe start with you, Dean, and then go to Kishor and then Mohammed.
Sure, it's a great question. I think that it's indication specific. So we do work with clinicians who look at using AI to develop digital biomarkers, to do image analysis. And then certainly in that space, I think there needs to be a fair amount of high quality data to drive the development of better ways to diagnose, especially longitudinally, which means you have to keep updating these data sets. But I think that for therapeutics, our goal here, right, is to take a patient biopsy, design the best possible combos for them within a few days off their biopsy, because there's so much heterogeneity there, et cetera. And then after we design the combos and you dose the combos, it's important to note that things like drug synergy are dose dependent, time dependent, and patient specific. Right, which means you give a combo which maybe was synergistic or good for the other person, maybe not so good for this next person, but it doesn't mean it can't be good for the person, but you may have to do some dynamic dose changing. To do that, the reason we don't use large amounts of data is because of such variability that exists patient to patient, which everybody knows, right? Any clinician will know that patients are different from each other, but we are different from ourselves over time. Right, so it's the perfect example of a patient gets recruited into a trial, they're given a combo, that combo does not work for that patient at clinical dosing, but then if you drop the dose of one drug at the right time, suddenly the combo starts working for a patient, and we see this. And so if we were to aggregate the data that we have on hand, a lot of that is missed. Right. And we can't implement that clinically without prospectively acquiring the data for just that patient and that variable dosing. So it's a longitudinal data set, but it involves variable dosing with corresponding efficacy and safety. And so that's why I think for interventional applications, treatment applications, I think it's important to ensure we acquire data in the right way, not just large amounts of data. And it's important to involve all the stakeholders in this. We often get asked a question, how well do clinicians pair with your platforms? And we say it's a lot more than them taking the recommendations and then using it if they believe it. They're actually at the tip of the spear. They actually acquire the data in the way that will be needed for them to then act on the recommendations. Right, so it's full circle. And it's very small amounts of data, but acquired in a very specific fashion.
Yeah.
So
recursive.
I'm okay. I have a different take, but I think it's because we're in a different approach in space. For us, it's about the quality of the data. So we do use a huge amount of data from lots of different databases and data sources. But I think what we're always concerned about is the quality of the data, where we get it from, and doing a quality assurance on that data. And so we have collaborations with folks that have those large data sets. So we do use a large amount of data, but we're always worried about the quality of that data because it can be misleading. And the other thing is, this is a fluid space. I mean, it's like, it's like a child, right? When, your outputs are only as good as your inputs, and the information is continually changing. I find how our society has attacked science over the last 2 years, but we all know as scientists that we follow the results. We try to test a hypothesis. We get our data, and whatever data tells us, then we go in that direction. We don't try to prove results. We don't try to prove a hypothesis. We try to test a hypothesis. And so when new information comes out and we have to change our ways of thinking about a problem, that's good science. But our general public doesn't see that. And so when our leaders make changes about COVID-19, they think that we're a bunch of flakes because we keep on changing our minds. Well, that's not true. As new data comes out and new inferences and new information, we change because that's what the data is telling us. And so it's the same way here. We do use large data sets, but we are also very cognizant of the quality of the data and the fact that the data is changing as we get new results and different things. How do we want to deal with it? Give you a specific example. Omicron replicates far faster and in the nasal cavity, as opposed to the original virus that replicates a little bit slower and deeper in the lungs. So our initial therapy was going to be a 10-day therapeutic study in patients, but the vast majority of the patients we're now treating are all Omicron, and they have a much quicker half-life of replication and symptoms. We had to change our whole protocol to fit that type of patient. So that's an example where, as new data comes in, we have to understand that data, we have to apply that data, and we have to make sure that that's high quality data.
Okay. Mohammed, have you been able to unmute yourself?
Yeah, it works for me now. Thank you.
Great. Do you want?
Yeah, I first wanted to thank both the speakers for their great talk. I really enjoyed both of them. And about I think I want to follow the same comment that when we use machine learning, obviously we need a huge amount of data set for training, and of course those data needs to be reliable, and we should make sure that they are qualified data that we can use for our approaches. But yeah, it's definitely there are some algorithms, like we have single shots algorithm in machine learning that doesn't require huge amount of data, but they're normally pre-trained on other data set. For example, if you want to apply if you have a bunch of images from cancer that you want to look at them, you can use a pre-trained imaging neural network, convolutional neural network, and apply that to your data set. So that means that then you need less amount of the data to be able to train your system.
Yeah, the efficiency of the data is more important than either quantity or quality. You had a great slide, Kishor, on what AI allows you to do, which is basically speed up that sort of R&D to applied process. And we just talked about how it's quality and not just quantity when it comes to data. And I'd like to ask each of you, again, start with Dean, then go to Kishor, and maybe then Mohammed. So we know what the upside of integrating AI and machine learning into Nano is. What should you be looking out for? What are the kind of pitfalls that AI brings to your work which might not have existed previously?
Yes, I think that what we do, certainly we develop combos and things like that, but at the heart of it, the bulk of what we do is at the clinical decision support space, right? I think right now we've probably got about, depending on what's in interim reporting, what's active, anywhere between 5 to 10 prospective trials happening in the CDS space. And I think that one of the biggest challenges, I'm actually going to take a step away. Of course, the clinicians need to understand how these recommendations are being made. And in order for them to have final sign off on whether they drop a dose, dynamically dose, or whether they pick combo number 2 or combo number 8. And the thing is, we've developed protocols for that, right? Before a clinician will pick a combo to give a patient, they're going to look at the positive, negative predictivity and what regimens the patients have been on before. They're going to assess the dose level, whether it's safe, all of that. I think one of the challenges now is how do you actually implement this in the clinic at scale? And so that's specifically why we brought in a team of behavioral scientists. We brought in people from industry. We have senior management who's also with clinical background, who runs trials for one of the biggest pharmas in the world attached to our institute right now. And we don't talk about AI digital from a technical perspective. He asked me a simple question. How long does it take for you to come up with a recommendation for a patient, right? And I told him, 5 years ago, it took 30 minutes. Today, it takes 5 minutes or less. And that might sound good, but it's actually too long. If you look at clinical workflow, if a doctor's got 30 patients a day, 50 patients a day, and it's 30 minutes per patient, it's a non-starter, right? A lot of great technologies were never implemented in the clinic because they couldn't be integrated into the workflows. And we're actually seeing that with even completely automated workflows, even where the clinician just has to look at the information, decide whether to go with it, but they do have to go backwards to go and see how that recommendation was made, and then it takes too long. And so I think the thing with AI in being implemented in a clinical intervention space is that there's a lot of work that has to be done on the behavioral sciences side. So we conduct interviews with doctors, nurses, pharmacists, caregivers, patients, all of the above. And I'll give one example. We had a case where we had recommended a lower dose for a patient. And the pharmacist on the team had mentioned, hey, given the way these drugs are incremented, it's actually going to be more pills. It's actually more pills at the lower dose. But if we slightly give a higher dose, it's actually less pills. This particular family, this patient has issues with adherence. So if you want to give the lower dose, that's fine. But if they have issues with adherence and they don't actually take their medication, it doesn't actually help them anyways, right? So there are many aspects of this workflow that we focus on to make sure that we can robustly implement this at scale. And so for us at Wisdom, we do integrate a lot of outside expertise in other domains to ensure we can deploy.
Kishor, what do you look out for with AI?
Yeah. So to answer your question, I'm an old school pharmacologist, pharmaceutical scientist. So I see artificial intelligence as an important tool, but I don't see it. I also don't want it to be fool's gold. And so, like I said, there are lots of people that asked us, are you going AI right to the clinic? Well, absolutely not. I mean, we're using AI to help us predict lead candidates, lead combos, lead therapies, which then we still have to validate by classic mechanisms and classic drug discovery and development work, cell work, formulation work, pharmacokinetic work, animal work, and ultimately proper clinical studies. So I think the pitfall for AI is that we don't get caught up in that it's the end all be all, but it's an incredibly predictive, helpful tool and helpful prediction to get us closer to nirvana and to something that we might want to have. One thing I will mention, I will tell you this: I've trained over 100 graduate students, postdocs, and so on, who most of them work in big pharma, make a lot more money than I do. Many of them now use they use AI all the time in big pharmaceutical industry. One of the areas, and Emmanuel Ho knows this very well, is an area called pharmacometrics. And what that is they use physiologically based pharmacokinetic analysis to be able to predict drug disposition, using AI platforms. And then based on that, then they can decide what is the dosing and what animal studies they need to do. But they still do the animal studies. So I think the lesson and the thing about AI is the output is only as good as the input. And you've got to be very careful about that. And I agree with my colleague. He made an excellent point, my colleague from Waterloo, that it is not only, you know, the amount of data, the quality of the data, but the efficiency of the data. And in fact, in pharmacometrics, the efficiency of the data is actually very important. So to me, that's the pitfall. That's the thing we have to be careful of.
And there's lots of cases from outside your fields where AI-based data sets, you know, in HR hirings take you down the wrong path because they're based on decisions that were wrong in the first place or discriminatory in the first place.
That's right.
Mohammed,
That's right.
Yeah, Mohammed, anything you would like to add?
Yeah, before I wanted to add anything, I saw Emmanuel's hand has been up for a while. Do you want him to go first?
Yeah, I was going to go to him after we'd ended
sure.
this round. I've seen his hand.
Okay, yeah. So I just wanted to add that you know, these quantitative, you know, approaches, if they're grounded in relevant experimental data, then definitely can provide a rational means of determining the, you know, optimal combination of different treatments and modalities, for example, and they can provide insight into, you know, into how do we target, for example, a specific disease like, you know, SARS-CoV-2, as was discussed. I think there are, if I'm not mistaken, there are over 70, you know, AI-associated medical, you know, devices approved by the US FDA already, and they're more like, you know, oncology-related fields. For example, that's the field that I am most interested. And that means that, you know, AI has been, you know, involved in many, you know, these, you know, medical activities, in particular oncology and other, you know, medical conditions. But one thing that I wanted to add is that, I mean, there are also approaches that we can try to interpret, you know, what we get from the AI. For example, this is not just a black box. Sometimes people look at, you know, machine learning and AI as just a black box that you give inputs and you get an output. There are also some ways that, you know, we can also combine them with mechanistic approach. We can look at, you know, interpretable, you know, approaches and try to understand the underlying mechanism. For example, if a combination works for a patient, or if a patient responds to a lower dose, what is the underlying mechanism for that? And then that would help us to understand better, you know, how we can extend it and how we can use it, you know, in for different patients.
Yeah. Emmanuel, you have your hand up.
Yes, thank you so much, Rohinton. Maybe a more specific question related to nanomedicine, probably directed more to Dr. Ho. I'm just curious because, you know, based on today's discussion, a lot of work is focused on applying AI to optimizing drug combination. Have your group done any work on optimizing or using AI to optimize the nano formulation itself? I'm just curious if looking or even combining the optimized, you know, drug formulation along with the actual drug itself, would that actually improve significantly, or have you had a chance to look at that? I know, you know, even for formulation scientists, it's a lot of work to optimize formulations themselves, right? You change one lipid, one polymer, you know, that can have a significant effect, but there's so many different types that you can use. I'm just curious if you have any comments regarding that.
Yeah, it's a great question. So, you know, we ran a study a few years ago where we, because there's been great discussion about inputs and outputs. And so for us, our optimization approach, the inputs are drugs, their doses. And in this case, the nanoparticle itself is an input. And this can be expanded into thinking about other ways of how you tune nanomaterial properties, et cetera. These can all be treated as inputs, for example. But in this case, we took 4 different drugs. And interestingly, some of the drugs are very similar in structure, right? So, and we had them unmodified by Nano and also modified by Nano, right? So the Nano is added as an input. And what's our objective? Our objective here is we took a range of different cancer cells and we said, kill as many cancer cells as you can. And then we took a range of control cells and we're like, keep as many of these alive as you can. And so when you take these therapies with or without Nano, and you know that some of these drugs are very similar to each other, one would think that, you know, maybe the best you can get out of this is an additive effect, right? If you take 2 drugs, 3 drugs, they're kind of the same, you add them together, it's really going to be additive. And because we wanted to kind of go after the notion of synergy here. Synergy tuned by with Nano, without Nano, drug, good drug, bad drug, drugs together, all of the above. And we found that if you combine Nano with this drug, Nano with that drug, The third drug, no nano, because we interrogated that whole spectrum of possibilities, you could actually find synergy that existed at specific correlating doses as well, right? So instead of saying, I think nano with this drug is good, nano with that drug is good, actually nano all the drugs, right? Because it'll help retention, for example. We're very agnostic, right? Because you do have to resolve the dose and the drug at the same time. Because even as we say in human, you tune the dose a little bit without changing the drugs, hugely different outcomes, right? And so what we found was in nano right now, the goals are, how do you tune material properties to get a release profile that you want? What drug should you load in the particle? Drug 1, drug 2, siRNA, aptamer, small molecule? Do you add a targeting agent? Yes or no? If you add a targeting agent, you probably will get more drug to the place that you want. But when you end up treating a person, do you really want that much more drug at a site for a specific person at a specific dose with these specific drugs added? We don't know. That remains to be seen, because certainly there are cases where you nanofy a drug and, early death can still happen, right? Even though these nanoparticles are supposed to reduce toxicity. So we take a very agnostic approach. Tell us what the inputs are. If the inputs are lipid composition like this, lipid composition like this, polymersome composition like this, these are the drugs we get to play with. We will treat that as a population of inputs. And then we will mechanism agnostic, even disease model agnostic, design the combos that are optimal for that. And it's not purely predictive, because you have to actually run the experiments driven by the optimization to prove, right? So it's not like we predict it's going to work, we run the experiment, hopefully it works. We're actually running prospective experiments. We're essentially crowdsourcing the disease model to tell us what the optimum formulations are. And in that study, it was optimized nano formulation, right? If somebody else says, yeah, I need to better tune, different composition of the material itself, plus pick the right drugs, we'll just treat those as discrete inputs and optimize for that too.
Thank you.
Do you want to add anything, Maha?
Yeah, just a quick comment. I wanted to say that, when, for example, you think about the effective chemotherapy, there are many ways that it's inefficient. We have inefficient drug delivery to the solid tumors. There are, various barriers, physical and biochemical, various mechanical, barriers. But if we think about it, when you use, nanoparticles, basically, as Dr. Ho mentioned, there are different release profiles, for example. Yeah. So think about it that you want to combine a few drugs, and then even 2 drugs, that you have different many ways that you can combine these together, because there are different release profile, there are different timing, there are different sequencing that you can use of them. And the way the AI can help is that, if you know what is the best release profile. I mean, I'm thinking about it as like the other way of the story. If I know what is the exact release profile that I need, and how much dosage is needed at this particular time, and what is the best sequence for for the chemotherapy to be effective, then I can think about how to build that nanoparticles with this specific release and this specific dosage at this certain time that is needed for this particular patient. So I mean, it's the same thing that Dr. Hu mentioned. I'm just saying that you can also think about it rather than which polymer I'm using. What is the characteristic of this nanoparticle that is needed for this particular patient, and then I can look over the library that exists for this different polymer and see which of them will give me that design characteristics.
Yeah, if I could jump in, that's an excellent point, because at the BC Cancer Agency in Vancouver, Dr. Marcel Bally and his colleagues are doing that in cancers already. And what they do is they look at the pharmacokinetic ADME principles of the 2 drug combination that they're looking at, understand what's the optimum dosing regimen and targeting and timing of the dosing of the 2 different drugs, and then go back and develop a drug delivery system that will optimize the pharmacokinetic properties of those 2 drugs for an optimal effect. So if we could use AI to help us tell us what is the optimum combination, then what you can do is then develop a drug delivery system that gives you that optimal pharmacokinetic profile. And in fact, we're working with Marcel with our AI to do that. Though drug delivery people have been doing that for years without AI, but I think the AI will definitely add to that. I mean, so I think that that's a very elegant approach and can be very helpful in helping in drug delivery.
Manu, do you have anything before we go to Wojciech? He has his hand up as well.
No, I'm good. Thank you.
Okay. Go ahead, Wojciech.
Two of you actually mentioned about the quality of the data. Can you explain us what the quality of the data means? How do you qualify good and bad data? And what do you do if you don't have a good set of data?
Dean, I was hoping you'd go first. You're more of an expert than I am.
Okay, I'll just very quickly say, I mean, and we have a small data. And our data is prospectively acquired. So we first of all have to set our boundaries for what that data calibration is, right? And as I mentioned earlier, it's a specific calibration regimen: low dose, high dose, medium dose, low, high, medium. We build that into our trial design. And then we have to establish a marker that corresponds to the variable doses that we get. In terms of our quality, we need a minimum, for example, of 3 data points, right? Each data point is 2 things: dose and efficacy. But we get more than that, okay? Just because whenever we take on a new cancer, if it's colorectal, if it's gastric, whatnot, we tend to get at least one, usually more extra data points, which it doesn't mean we have to get even more dose variability. It just means we need a couple extra data points at the same dose to see how much drift there is in the marker on its own at the same dose, right? And that tells us a few things. If there's a biomarker spike in some patients, if you give them a dose of a drug, you take the next reading, their tumor markers are through the roof. And then you're like, wait, is this resistance, recurrence, progression, or is this just a spike because, you know, it's a natural release as tumor cells die and then it comes back down? We cannot act on the dosing until we figure that out, right? So for us, the quality is if we look at a data set and we did get the requisite 4 or 5 data points that we want, right? And we're still not confident that we're in the middle of a biomarker spike or not, we can't act on that data yet, right? And this takes discussion with the clinicians where they're like, given that this is settled down, you know, with no escalation in dose, we're confident that this is a spike and it's come back down to earth. These data points are dependable. We can move on. It sounds like a lot of discussion, but it's actually pretty fast because it's not a lot of data points to discuss, right? It's a very quick conversation and then we move on. So it's for us, it's the actionability of the small data set before we can implement the intervention.
So so the answer to your question, Wojciech, about large data sets and knowing the quality of the data set, the 2 approaches that we have employed are the following. If we're getting, let's say, ADME data, large data sets of ADME data for drugs that we're going to put into our AI, we try to get it from multiple sources of reputable people who are doing world-class work. But we get it from multiple sources and be able to see if they're consistent, if the data is consistent on, absorption, metabolism, distribution and excretion of a drug, and making sure it's so on. So, for instance, the drug unit at the University of Washington in Seattle is one of world renowned in large data sets for ADME, right? And so we collaborate with them to get their data set. But UCSF down the street also has a similar data set. So what we try to do is we get multiple data sets for the same things that we're looking at and comparable to make sure that they line up. And that's how you do it. So, you have to trust your partners in terms of those large data sets, and you work with reputable organizations that are experts in that specific area, which makes it which makes our task also what makes our task, I think, even more difficult is when you're trying to input data sets for different aspects of a drug or a parameter, like drug-drug interactions, or ADME, or drug toxicity, or formulation, or physical chemical properties. The hard part is to integrate all that in a common platform, which is what our computer science graduate students are doing at LSU. That is actually one of the bigger challenges than making sure the quality of the data is of high quality, because we get that from reputable people that we know have got they've done world-class work, and we get it from multiple people where the data is consistent for a specific drug or whatever we're trying to look at. If you're trying to do grassroots work, I think what Dean is saying is correct. For smaller data sets, we don't work in that smaller space. We work in those larger data space. So we're really at the beck and call of those groups that work on those big data sets for those specific parameters that they're investigating.
Mohammed, would you like to add anything to the question of quality of data?
Yeah, I think the interesting and absolutely valuable comments were raised by Dr. Hu and Kishor. I just wanted to add that many times we have noise in data, and for example, sometimes we also have incorrect label that we have to be careful with them. But as was discussed, one important thing is that you go, for example, you do a cell culture experiment in one lab and another lab, you just do two different cell line, different passages, and so the result might be different. So when you combine these two together, you're bringing some sort of inconsistency. So you have to be careful how we are pulling this information data together. And sometimes, for example, in clinic, for example, we were looking at immunotherapy response for patient with breast cancer, and we had like hundreds of patients, but 30 of these patients had missing data. For example, some of these data were not collected basically. So there are some ways to carefully pull the data together, and also there are some ways to trying to estimate the missing data for a given patient that you can use using machine learning algorithms, and sometimes that again we have similar data. For example, we're looking at SARS-CoV-2, for example, genomic data, and you have over 200 million data sets, but when you don't download them, so many of them are just repetition of each other, but they're not significant mutations from one to another one. So you have to be careful how you collect valuable and effective and qualified data from this sort of data set.
So let's go back to Wojciech, and then we have a very interesting question in the chat, which I'd like to ask Dean and Kishor about as well.
I think we can start with the question on the chat because it was before me, so I'm absolutely happy.
Oh, okay. I thought you had a follow-up to your previous.
No.
It's directed at Dean, but it's really to both of you. Is how do you see the scaling up and moving, if I could call it, to market? And let me just add an addendum to that, because vaccine equity is all the rage currently. Is what are your policies or approaches to IP and eventually pricing?
It's a great question. So one thing that's quite exciting with what we're doing at our National University Hospital here is we have a powerful AI platform that has been integrated. It's operation scale, right? Where we can make data available fairly quickly across all indications. And our objective here is when a patient has been naturally calibrated. By the way, we talk about dynamic calibration. A lot of that actually happens as part of standard of care already. Transplant patients, endocrinology, and even in some cases, oncology. And when that happens, the system will, and certainly hypertension as well, which is a major issue in the region. And so our objective here is when the patients have been calibrated sufficiently that a button will pop up, right? And it'll say curate. And what's going to happen is this will automatically allow us to provide decision support on the side for a patient based upon natural calibration. This, of course, is in parallel. This is all the approvals in place, with the approvals in place, where we can then implement this. And of course, we'll have trials going on as well. So that's the operational side of being able to scale this and implement this. And we've been working on this for a while now. With regards to scale up from the clinician's perspective, that's why we integrate the behavioral sciences side in order to understand how we take that recommendation workflow and build it into the standard patient treatment workflow. And that's ongoing. A lot of that is qualitative research. So we understand what the concerns are, what the challenges are, what the opportunities, what they like. And when it comes to interfaces, even, we're working with design faculty who specialize in interaction design, UX and UI, to make sure that this can be scaled up from a usability standpoint. From an economic standpoint, that's also happening in parallel. As I mentioned earlier, given how the payer systems work across different geographies, there's going to have to be unique development across the globe. But at least for us here, we are currently running analysis on all of our trials of how much money we are saving the patient as well as the healthcare system. And as I mentioned, our very first hire is actually from the reimbursement payer side. And we actually don't initiate any programs until we get feedback from him on the KPIs we need to meet so that we can scale. When it comes to things like economics, whether it's a startup or whether it's public benefit, we have all of that. We examine all of that. For our COVID work, it's actually purely a public benefit platform. We work with our clinicians here to pinpoint the drugs they are interested in looking at to then help them better prioritize from that. Purely public benefit, it's actually for the globe. We're on the phone with Taiwan yesterday. We've spoken to University of Florida not long ago, purely for public benefit. For other areas, it takes substantial resources to scale up, right? And before we do that, of course, we can go in and spin something off and try to do a raise. But in order to scale up N of one type healthcare, there are substantial healthcare economics and logistics and infrastructural considerations that are needed. We try to address those early on. And that's why our institute looks at this before we initiate any project, actually. And then we start to look at the economics in parallel. But I think we're doing pretty well. I think in terms of how many patients each person can handle from an optimization perspective, cost savings, computation needed, which is actually not that substantial because we're doing small data. I'd say we're going to start with certain oncology indications and then move now towards hypertension next. We have a fairly disciplined plan to scale this up by indication. But thanks to our community, which again spans technical, policy, regulatory, payer, I think within the next 2 years, we're going to see a pretty big jump in the number of patients being served by this space.
Kishor?
Okay. All right. Couple of things. So number one is I'm the co-founder of the Neglected Global Diseases Initiative at UBC, and we have a global access policy, which means for developing world indications, we develop drugs at or below cost for the developing world. One of my technologies is part of that program that was funded by the Gates Foundation and is going into phase 2 clinical trials. And we're not trying to make money. It's mostly cost recovery to help patients. For SkyMount, part of the reason why we've decided to work with repurposing drug strategy is our 2-drug combination is our generic drugs that are very cost effective, that are cheap, that are very accessible in the developing world. In fact, one of the drugs is very well known and very well used for another indication in the developing world, and it's pennies a day. And for SkyMount Medical, it's a humanitarian mission. We purposely picked drugs that are generic, that are cost effective, that are accessible, that are easy to use, that can be administered orally. And in fact, we want to get to people who don't have the privileges and so on. Vaccine equity is a very important issue for us at UBC and at SkyMount. We just co-sponsored a symposium on vaccine equity in November at UBC, discussing exactly this issue, and in fact have extended that to oral therapeutics. So that's how we feel about that. And UBC was the first in the world to have a global access policy, the Global Access Initiative in 2009. And we worked with an organization that came out of Yale University from students called UAEM. You may be familiar with them, Universities Allied for Essential Medicines. This is a group of students, and they have chapters all around the world with the idea of developing drugs at or below cost for developing world indications and for things like COVID-19.
Very inspiring indeed. Mohammed, anything to add?
I just wanted to use the opportunity and acknowledge the work of one of our colleagues, Alex Wong. He is a professor and clinical researcher at UW, and this is the same policy in our university. So I think he developed a deep learning algorithm for X-ray imaging for COVID-19 patients. And the data is available. It's open source release of COVIDNet, CXR, and that's the idea is that would be the algorithm would be able to pick up patients from the X-ray imaging. So I mean, this is the same policy that we have been following. If there is something going on, will be open source and available globally for anyone who wants to use it.
Yeah,
and
we wrote a paper about it in 2009. I want to go back to one thing. I didn't fully address Wojciech's question about data, and I mean, it's always very difficult to making sure you have high quality data. But at the grassroots level, as we all know, I'm a basic scientist, is you look at the methodology that people are using to get their results, and then you got to see if you can reproduce it. And really, it's only reproducible data that that from multiple different labs that you can use, that you can say, well, we have some confidence that this data is real, as opposed to it being goofy. And that's a difficult task. And one of the graduate students at LSU in computer science that I'm mentoring right now is actually doing that kind of quality assurance work. And to be honest with you, it's very hard work, because he sees different labs with slightly different results, but then their methodologies were set up differently. They didn't do the proper positive controls. They had different conditions and so on. And so that is very hard and tedious work, but we try to achieve that. So I hope that gives you further clarity on your question. Your question is an outstanding question. You're absolutely right. It's how do you determine the quality? And it's painstaking, and it's going to be part of this young graduate student's PhD thesis. And he's not a happy camper right now, because he's having to, it's like needle in the haystack, right? He's going through these huge data sets to do that. And it's easy for me to sit here with my gray hair and tell it all this. He's going through the hard work, and I'm going to give him a shout out. His name is Adam Best. He's an outstanding graduate student, and he's going through that quality assurance right now. So anyway, just further comment to your comment.
I suspect, Wojciech, this is the last question and the last round of discussion, so that I can hand it back to Emmanuel
Yes.
a couple of minutes before closing time.
Look, I want to ask something which I believe is quite important as well: regulations. Obviously, your work is looking into saving lives, reducing toxicity, etc. But we can very easily envisage AI being abused to create a new illicit drugs, cheaper, more effective, more accessible, or in sport doping. Can you please provide some comments on this?
Well, I'll make a comment, and I'll then let Dean. That's an excellent point. The FDA has done a number of workshops on AI ethics, and in itself a discipline. It is itself becoming a huge discipline. I think there's a research chair, I believe at LSU, who is his expertise on AI ethics and AI abuse. And every week I see the FDA as well as Health Canada, I'm here in Canada, doing workshops and discussions around exactly what you've brought up. Because, in classic fashion, the science has gotten ahead of the ethics. And now ethical concerns have come up for all the things that you've just brought up. So that is a big concern. We have a submission into the FDA right now for one of our therapies, and they have some expertise on the ethics of the AI that we've used, but I think it's behind. I think it hasn't caught up to what's happening out there in the AI area. So that's a very good point. I do see now people who are doing expertise in that area. There are workshops in that area. There are institutes that have people who are doing that. That's their research on the ethics of AI, how to control all that. So I think more needs to be done. I think we're only at the tip of the iceberg, and unfortunately, there are going to be bad actors, and you have to find a way to control those. I'm sure Dean must know from all the great work that he's done and his institute's done some of the things that he faces.
I'll just quickly add. So I currently serve as a subgroup lead in the WHO's AI for Health Regulatory Considerations Working Group. And I think that something that's essential is to substantially increase the dialogue between regulators around the world, and then because there are unique constraints as well as opportunities that face varying jurisdictions and geographies. And a part of, and we have been doing that. We've had regular interactions to kind of look at lessons learned, to look at policies that have worked. And this working group includes ethics lawyers, those from regulatory agencies, tech providers, clinical trial design expertise. It's all in there. And some of the lessons learned include the importance of tech developers engaging their regulators where applicable and appropriate to help advance the regulatory workflow and resource landscape. The example I'll give is us here in Singapore, where I think we're privileged in the fact that if I want to develop a clear path to getting a platform, an AI platform submitted for regulatory clearance, I need to have discussions with them. And they are so efficient at having these discussions while maintaining, the highest levels of rigor with review. I've had cases where I wanted to deploy a first-in-kind trial, and I contact them on a Monday, and I'm meeting with them on Wednesday, maybe Tuesday, right? I've had cases when COVID happened, and I needed a guidance from them, and I called them at 11 in the morning, and by the conclusion of that call, plus 10 minutes, they sent me an email with the guidance so that I could then interact with my senior management to get a project initiated. I would think that level of engagement is nearly unprecedented, right? And in fact, in some of our reports, we're mentioning that this type of engagement can save lives, right? So back to your question about, when it comes to the major challenges of deploying AI, whether it comes to, to sport, to athletics, and to other areas, I think first of all, empowering regulators with kind of the landscape, the state of the art on a frequent basis will further allow the regulators to empower the whole industry as it moves forward. Right. It really is. It really needs to be seamlessly integrated. And then this will help the regulators develop resources so that the tech developers have a faster, clear, more compliant way to advance their technologies as well. It's really about dialogue with your own regulators, and then dialogue between regulators informed by all the outside expertise that's needed in such a dynamic field. And part of the privilege of being here in Singapore and working with our regulators, as well as part of the privilege of being part of the WHO working group, is that we're able to constantly learn, right? If we're willing and admitting that we have to constantly learn, we can prevent the barriers, and then we can move beyond that to catalyze faster progress for the field.
That's very heartening, but I should also say that that's also very Singapore. And I suspect the big thing now is that algorithmic accountability barely exists. More than half the countries in the world don't have meaningful privacy legislation, and on and on and on. At CG, we have a huge sort of depressing set of analyses on big data governance. Mohammed, I'd like to sort of end with you and then have us wind up with Emmanuel.
I think the main points were already discussed. I don't want to add too much. I think that, as you guys mentioned, I think having all this, personal information in various, discipline and how we can deal with that. It's a big issue. It's a very important, thing that need to be considered carefully. And I know that in Canada, globally, people are looking into this carefully to have, specific policies that, how this, should be addressed.
Great. Look, I want to thank all three of you for a very inspiring discussion. I know this will continue into tomorrow and then your big meeting in June. It's been valuable and what a great start to the network. Thank you all. And I think at this point, I will turn it over back to you, Emmanuel.
Thank you so much, Rohinton, for doing an amazing job in moderating today's session. Again, thank you to all our speakers and panel members. And most importantly, thank you to all the attendees for making today's session a huge success. I personally have learned so much regarding how digital health, AI are really transforming medicines, nanotechnology, and really how it can be used to create a healthy and sustainable future. So, without going too much further, I just want to remind everyone, tomorrow is day 2 of our event. Tomorrow's session will be focused on nanomedicine. So feel free to take a look at our website for additional information as well as registration details. And thank you very much, everyone. Please enjoy the rest of your day, and hopefully we'll get a chance to see you tomorrow. Thank you and take care.
Thank you.